Reproduction in viruses. Viruses are tiny structures which can be seen only by electron microscope. We now remember this information from chapter 1. They represent a connecting link between living and non-living things because out of host cell, which can be human, animals, plants, or other living things, they behave like living organisms, but out of host, they don't show any properties of living things, so they are non-living. They are able to grow and reproduce inside the living cells of other organisms, but cannot survive in external environment. So we ask question, why? Because the viruses have no cellular mechanism to reproduce independently. Information about reproduction is obtained by observing a type of virus attacking a kind of bacteria called E. coli, Escherichia coli. It's name of bacteria which is infected by virus. The virus which infect that bacteria, we call it bacteriophage. So most of information about reproduction of viruses we gain from information about bacteriophage. Bacteriophage in structure look like a spaceship, the head, middle piece, and tail fibers. So in this capsule-like structure has a protein coat which includes a piece of genetic material, can be DNA or RNA according to kind of organism. A tail, a tubular structure, and tail fibers. Virus use it to attach the surface of host cell. So they have no cytoplasm, no ribosome to produce proteins or other cellular structures, so they cannot reproduce independently, but they can use the parts of other cells and they can reproduce in host cells. So reproduction in viruses can be in two stages or two types, Lytic cycle and lysogenic cycle. First, we're going to study on Lytic cycle, which lyses, digest, or broke, or you can say kill the host cell. The first stage is attachment stage, when virus, which has spaceship, tail with tail fibers, the virus become in contact with bacteria. We remember structure of bacteria again from the chapter 1, covered by a capsule, cell wall, and cell membrane. The fibers, which are tail fibers, exist in the tail, stick to spatial position on the cellular wall of host cell. Host cell is a bacteria because we said this virus is bacteriophage. Attachment stage, virus attach the surface of host cell with fibers on its tail. Penetration stage, again, bacteria attacked by virus, which call it bacteriophage. Tail attach the surface by tail fibers, and the enzyme, viral DNA, and bacterial DNA. The enzyme found in the tail decompose the cell of bacteria in the region of adhesion. So there's an enzyme on tail of virus. It make a hole on surface of bacteria and inject its genetic material or viral DNA into the cytoplasm of bacteria. So we call it penetration stage because the viral DNA is penetrated or injected into the host cell through tail and through the hole which make on the surface of host cell. Biosynthesis stage, bacteria, after virus inject its viral DNA into the cytoplasm of host cell, the remaining part of virus are not necessary. So there are bacterial DNA and viral DNA in cytoplasm of host cell. When the viral DNA entered the bacteria in previous stage, it transcribed messenger RNA. There are two types of nucleic acids, means DNA. DNA and RNA. RNA like assistant of DNA, or DNA control the cell activities, produce proteins by what? By transcription, means copying of messenger RNA. So the viral DNA transcript messenger RNA necessary for construction of enzymes for degeneration of DNA and mRNA of bacteria. So the viral DNA produce enzymes. By these enzymes degenerate the DNA or genetic material of bacteria. Then the cellular mechanism of bacteria produce of proteins and release energy under the control of viral DNA. Viral DNA direct mechanism of host cell for the formation of nucleic acid and new viral proteins. So many viral proteins like this are produced in cytoplasm of host cell when after virus take control of cell and pieces of genetic material DNA. In biosynthesis stage? Simply we can say in biosynthesis stage, the viral DNA which is injected in the cytoplasm of host cell in penetration stage produce enzyme, broke down DNA and RNA of bacteria, take control of cell and produce proteins and new viral DNA for construction of new viruses. In next stage, the maturation stage, again bacteria. In cytoplasm of bacteria, a lot of viruses are produced. So we remember in previous stage, pieces, proteins and DNA are produced, but in maturation stage, these pieces, protein molecules and DNA are organized in order to form the protein covers around the new strips of DNA. DNA around DNA, protein coats, tail and fibers. DNA, protein coat, tail and fibers. Viral DNA, protein coat, tail and fibers. In this method, it produces about 100 to 200 new viruses are produced. And this produced viruses are released in next stage, which we call it release stage. New viruses cause decomposition of the host bacterial cell. How? By again producing some digestive enzymes. These viruses are released in order to infect other disinfected bacteria. The process completely takes about 25 minutes. Means one viruses in 25 minutes produce 100 to 200 new viruses. But how? In a host cell. Because the host cells are broken down, digested or destroyed, we call the stage lytic cycle of reproduction in virus. In lysogenic cycle, attachment and penetration stage occur as in lytic cycle. Means virus again find a bacteria, attach the surface, inject its genetic material, but then the mechanism is a bit different. So bacteria, bacterial DNA, And viral DNA, the nucleic acid of virus incorporate with nucleic acid of bacteria. Not broke down DNA of bacteria, but cut and insert itself into DNA of bacteria. So each time bacteria divide into two by binary fission to form two new bacteria, and also it produces two new viruses, because these two cells, both of them are infected with virus. So anytime when virus decide, it can pass to the biosynthesis stage, broke down DNA of bacteria, take control of cell, produce new viruses, and release to infect other bacteria in lysogenic cycle, then can be converted into lytic cycle 2.